Part 3. Building the model, the difficult parts. So this is really about building the structurally variable regions and the sidechains. So structurally variable regions in the general case tend to be loops, and in the case of antibodies it's the CDR loops that we're interested in. And this is again that CDRL1 that we've seen previously. Inevitably, the structurally variable regions will be built with lower accuracy than the rest of the structure. Even when the length is conserved, they can adopt quite different conformations. But in the case of antibodies, this is relatively straightforward with the exception of CDRH3. Now, from the antibody lectures, you should recall that Kabat and Wu identified these complementarity determining regions, the CDRs, based on this sequence variability. So they produced this variability plot, and this was at a time when they had no structural information on antibodies. And they predicted that these three very variable regions would form loops, and that these loops would come together to form the combining site. Once the structure was solved, they were proved to be exactly right, and you can see in red, green and blue CDRs L1, L2 and L3. Now, the assumption at the time that they did this was that the conformations of the CDRs would also be highly variable, given that the sequences were highly variable. But Cyrus Choudhia, working in the 1980s, found that this really wasn't the case. So he examined about 8 structures of antibodies, which was all that was available at the time, and found that the structures of the loops were remarkably conserved even when you had length variability. So this was the point that I was making earlier on about alignment. And he defined then the conformations of the loops based on CDR length. Obviously, this red one doesn't have the same conformation as this blue one, although this base region is really quite similar. So his first criterion then was CDR length, and the second criterion was the presence of certain key residues, either within the loop itself or packing against the loop. Now, as I said, that was done with only about 8 structures, and I was actually working on antibody modelling at that time, and it was our feeling that it was unlikely that this would generalize when we had lots more structures. It turned out, however, that he was exactly right, and this was the final paper that he published on this topic in 1997, where at this stage we had about 50 antibody structures available, and really his rules and his idea was shown to be exactly correct. So here we've got a number of loops of different lengths, and you can see they fall into nice tight clusters. Let's look at a 10 residue CDRL1. This is again from this antibody called HIHEL5 that binds to lysozyme. And I've highlighted here the key residues. So we have within the loop an alanine at position 25, a valine at position 29, and a methionine at position 33. And outside the loop we have an isoleucine at position 2 and a tyrosine at position 71. And if we space fill those, it's not quite so clear, but these in blue are the 3 residues that are within the loop, and then in red and green the 2 residues that are outside the loop. So you can see that what's happening is that these 3 are coming together and forming the centre of a sandwich, where the slices of bread are these 2 residues on the outside, holding everything in place. And providing you maintain those key residues, you can vary the other residues without having an effect on the overall conformation. Now, his analysis showed that you don't have to have specific individual amino acids at those positions. There is in fact some variability. So we have to have the isoleucine at position 2, the alanine at position 25, but at position 29 here we can have any of the kind of key hydrophobic residues, valine, isoleucine or leucine, and this methionine can be replaced by a leucine, and the tyrosine at this position can be replaced by a phenylalanine. But providing you conserve these residues within those groups, you'll maintain the same overall structure. So this is just for the 10 residue CDR-L1s. The different CDRs and the different lengths all have their own sets of key residues. Now this is CDR-H2, and these two examples in red and in green are both of length 10, but this red one is clearly a different conformation from the green one, and that seems to result from a difference in this framework position 71, where if you have a long side chain here like arginine, you end up with this green conformation, and if you have a short side chain like alanine, leucine or threonine, you end up with the red conformation. So what all this means is that you can look purely at the amino acid sequence, and you can define a canonical class for each of the CDRs, so you know what conformation it's going to adopt. And that makes the building of these structurally variable regions in antibodies much simpler and much more reliable than in the general case of building SVRs. So what we tend to do, at least, is we will find the templates that have the correct canonical class, which we know from the sequence information of the target, and then we choose the template which has the highest overall sequence identity to our target structurally variable region. Now that brings us on to the problem of how do we join a loop onto the framework. Well, the way that we approach it is based on a method that was described for a modelling program created at Birkbeck some time ago called Composer. And what we do here is we take our framework, which is in red, and that's carrying on down below the screen, and we've got a loop here in green plus its overlapping region of framework. So what we do is we look at the 3 residues before the loop on either side, and we fit our loop to that framework using those residues. Now that brings us on to CDRH3. The problem with CDRH3 is that it doesn't obey these canonical class rules. CDRH3 is much more variable in length and in sequence than any of the other CDRs. It can go from 2 or 3 amino acids up to 40 or 50 amino acids. Most of them are around about 10, and 10 residue loops are generally modelled with reasonable accuracy. But when we get to the very long CDRH3s that are typical of species like cows, it's almost impossible to model them. So how can we set about modelling CDRH3? Well, one approach is to model by hand. We can start with a similar loop and try to modify it using molecular graphics. And that's rarely done now. It was popular in the 1980s, and the first part of my PhD was actually trying to do that and then proving that it really didn't work. The other approach can be described as saturate and screen. In other words, we try to saturate conformational space. We try to look at all the possible conformations for the loop, and then we screen those in some way in order to find the most likely true conformation. Now, there are two major approaches to saturating conformational space. The first is known as the ab initio approach, and that really does attempt to search all possible conformations. And when I say possible, I mean energetically favourable. And that can be done using true conformational search techniques, so there's software called ConGen that can do that, or using molecular dynamics type approaches. The second approach is known as the knowledge-based approach, and that uses a database of loops from other proteins, not necessarily homologs in general, and of course for the case of antibodies, not necessarily antibodies. Now this, in principle at least, allows a more intelligent sampling of space, because we know that the conformations that we're looking at have been observed in other proteins. That said, there have been some papers fairly recently that have suggested that antibody CDRH3 loops adopt conformations that are nothing like loops seen in other proteins. So it may not be the best way to proceed. Thirdly, we can also combine those two major approaches, and that was work that I did some time ago, building the bases of loops using a knowledge-based approach and the tip of the loop where it's most likely to interact with the antigen using an ab initio approach. Now, however you've generated these conformations to try to saturate conformational space, you then need to screen them to find the most likely true answer. So the first approach is an energy-based approach. Obviously, we always screen out for clashes first of all, so if we're using the knowledge-based approach, we might be looking at loops that simply cannot join onto the framework properly because they clash with other parts of the structure. So we would screen those out. But also we might look at the conformational energy, including the interactions that the loop would make with its surroundings. We can do sequence identity methods, so we can calculate the identity of the parent and the target loops. We can use sequence similarity methods, where we do the same sort of thing, but we use some sort of scoring matrix for amino acid similarity. We can use key residue approaches. Now, we can't do that in the same way that we can for the other CDRs, but there is a set of rules known as the Shirai rules, which have been generated for the base region of CDRH3, which tends to adopt one of two alternative conformations. Or we can use some sort of automated analysis of key residues, which is an approach that was taken in that Composer software that was developed at Birkbeck that I mentioned a while ago. Or we could use solvent exposure based methods. So loops tend to be relatively compact. They tend not just to be sticking out into solution. And also hydrophobic residues are likely to be buried and hydrophilics exposed. So we can look at those sorts of things. And of course we can combine all these different approaches together. And one way of doing that, of course, is to use machine learning methods. And again, machine learning methods have been applied in terms of looking for favoured contacts between residues, in other words, predicting how far apart residues in a loop are going to be. Once we've built the structurally variable regions, we need to replace the side chains, and this may need to be done before doing some of those evaluation methods. For example, looking at energy, looking at exposure of hydrophobics, all that sort of thing would need to be done after you've replaced the side chains. So the way that we approach this, the most basic approach, is known as the maximum overlap protocol, or MOP. And essentially what we do there is simply inherit torsion angles from the parent and build any additional atoms using a standard conformation. So in this case, if we were replacing this serine with a tyrosine, we would put this C gamma position where the O gamma was in that serine side chain. And then the rest of the side chain is simply built in a standard conformation. If we were going the other way, so we were mutating from a tyrosine to a serine, we would simply cut off the extra atoms and change this from being a carbon to being an oxygen. The next step on from that is known as Minimum Perturbation Protocol, or MPP. Here we start with MOP and then rotate the side chain. In this case we have two torsion angles. We can rotate this one and this one in order to avoid clashes. But a better approach in general tends to be to use what's known as rotamer libraries. So when you look at the side chains of amino acids, there's always a preference for staggered conformations. So we can see this over on the right. This is a Fisher projection where this small circle represents an atom closer to you and the big circle is an atom farther away, and you're looking down the bond between the two of them. Now the atom at the back has the other atoms that are connected to it in staggered positions compared with the atom at the front. Now it turns out, in fact, that there's not just a preference for staggered conformations, but there is a preference for particular staggered conformations. So in this case, over on the left here, I've taken about 50 tyrosine residues completely at random from the protein databank, and I've fitted them on their backbone and then just shown you the side chains. So you can see there are three clusters, one here, and one here. But this cluster on the right is more populated and tighter than the one on the left, and the one at the back here has only got a single member. So there's a clear preference for this one, followed by this one, and then hardly any in this one. Whereas here I'm looking at phenylalanine, and in this case this cluster on the left is hugely populated compared with the one on the right, and the one at the back there are no members. So this program called Squirrel is able to make use of this type of rotamer library information, these preferences for particular staggered conformations, and it uses separate libraries depending on the secondary structure that the side chain is present in. And essentially what it does is to start with the parent structure, replace any side chains that need to be replaced, putting them into their most favoured conformation, and then it looks for any clashes and moves those clashing residues to second most preferred position, and it keeps kind of iterating around that until it's come to a solution where all the side chains are happy. The final step then is refining the model. And here we can use things like energy minimization or molecular dynamics. These treat the protein as a set of Newtonian springs. We have optimum values for bond lengths, angles, torsion angles, van der Waals packing, electrostatics. In molecular dynamics, we assign some sort of initial velocity to each of the atoms from a Boltzmann distribution, which is dependent on the temperature. And then what we're doing effectively is iteratively integrating Newton's laws of motion, and we're allowing the structure to rattle around. In energy minimization, we're simply moving atoms to an energy minimum. The problem with this is that it can only make minor movements, and it will tend to get trapped in a local minimum. However, if we've done all the previous steps properly in this process, at least with antibodies, energy minimization tends to be enough. Because the sequence identity is relatively high, the structures are relatively conserved, and all we really need to worry about is the splice of loops onto the framework. So energy minimization is generally enough to sort that out. In the general modelling case, we're more likely to have to use something like molecular dynamics and then take snapshots from that time series of the motion of the protein and energy minimize each of those snapshots to find the one which has the overall lowest energy. Having done all that, we then want to assess how likely the model is to be correct. Now that, for most proteins, is a case of using one of the tools that's available to look at whether residues are happy in their particular locations. In the case of antibodies, we don't really need to do that so much, because really the problem boils down to CDRH3. And there are now a couple of methods being developed to look at the quality of CDRH3 model, to look at its environment, to look at its energy, to look at its shape, to look at similarity with the parent, and so on, in order to make a prediction as to whether the model is likely to be accurate or not. So how good are the models? Well, in the general case, we can divide our modelling problem really up into about 4 bands. So when the sequence identity is greater than 70%, we generally have very, very high quality models. The RMS deviation may be as low as about half an angstrom, and that really is the same sort of quality as a crystal structure. So if you take 2 crystal structures that have been solved independently of the same protein, typically they will have an RMS deviation around about that sort of value. Between about 55 and 70%, again we get high quality models, and the RMS deviation is likely to be less than 2 angstroms, and that's really the sort of threshold that we generally look for a model being really good. Between 35 and 55%, things tend to be a bit more variable. Sometimes we will get very good models, but the RMS deviation generally is likely to be between 4 and 6 angstroms. Below about 35% sequence identity, models tend to be of poor quality, with RMS deviations greater than 6 angstroms. Now, AlphaFold 2 has changed some of those thresholds somewhat, but at the moment it is still a proprietary program and it's not available to people outside the developers. If we look at the antibody sequences that are available and compare them with the antibody structures, then we can see how many sequences we have templates for at each of these sorts of thresholds. So you see that surprisingly, even though the antibodies you think of as being relatively conserved, at least outside the hypervariable loops, the majority of antibodies actually only have templates that are in the range of 35 to 55% sequence identity. But we have a fair number above 70%, good number between 55 and 70%, and very, very few below 35%. Nonetheless, because of these special features of antibodies, the fact that it's only these variable domains that we need to model, the fact that structurally the variable domains are very conserved outside the CDRs, and the fact that, with the exception of CDRH3, the CDRs are relatively easy to model, we are generally expecting to get much, much better models than we would do if we're looking at the general case here. In conclusion, there are lots of different pieces of software for modelling antibodies. So one of the first automated methods was called ABM, and that was based on software that I developed during my PhD. WAM was another related system based on the same underlying code. So ABM was commercial and WAM was web-based. Other programs include PIGS from Anna Tramontano's group in Rome, Rosetta Antibody, which uses a rather different sort of approach. PIGS and ABM and our Abimod, which is our new antibody modelling software, are all relatively similar in their approaches and similar to what I've described. Rosetta Antibody is rather more complex. It uses methods that come from robotics for building loops. And then there are methods from Charlotte Dean's group in Oxford, methods that come from companies. This one is a protocol rather than software specifically for antibody modelling. It makes use of Modeler and another program called Looper. And this one from CCG is also a protocol based around the MOE software. And similarly, Schrödinger has one, which is a protocol based around 3 of their pieces of software. Now, all these methods get assessed in an experiment known as the Antibody Modelling Assessment, or AMA. And this is a little bit like the CASP experiment that's done regularly for general protein modelling. So the last of these was actually some while ago now, in 2014, and published in Proteins. Just to conclude, overall then, antibody modelling is similar in approach to standard comparative modelling, but it has a number of issues that make it a bit different. So first of all, we have to deal with the fact that we're always dealing with a light chain and a heavy chain. But on the other hand, we have the number of things that make the process simpler, because we're really just concentrating on modelling those 6 loops, and for 5 of them, these canonical rules from Cyrus Choudhury really seem to work very, very well. Which means that really the problem boils down to just modelling CDRH3, which turns out to be very difficult. So when the CDRH3 is shorter than around 10 to 12 amino acids, it's not too bad a problem, and we generally get reasonably good models. But once we get longer than that, it gets harder and harder.